Hello? Hi. Could you turn off the microphone? Hello, everybody. Thank you very much for coming to Novel Imaging Acquisition Method session. My name is Hiromasa Takemura, who is going to be a chair of this session together with Dr. Peter Bandettini. Maybe I have a few announcements. First of all, maybe if you have a question, you can either raise your hand here. Then there's one microphone over there. There's other microphone over there. Well, sorry, there's no standing microphone here. Maybe you can just pass around or speak loudly.
Or use the app.
Yes, alternatively, you can use the app. Then either way, it's fine, okay? So let's move on to the first presentation. So first speaker is Sebastian Dressbacher. His title is High Temporal Spatial Resolution Basal Review Differential Laminar Reactivity to Short Stimuli at 7 Tesla.
Thank you very much. So this is work I'm doing with supervisors Renzo Huber and Rainer Göbel, who is here as part of my PhD, and I'm very honored to present this work to you. So this is Charlie, and Charlie is a neuroscientist who uses high-field fMRI to look at layers and columns in the living human brain. And Charlie knows that the commonly used gradient echo bolt gives you a lot of signal, but it is heavily biased towards the large draining veins, which pool the blood from deep layers towards the surface and then away, which is an issue if we want to look at depth-dependent signals. Alternative methods have been proposed, like CBV-based imaging, which are then thought to be more specific to the microvasculature, and thus also the neurons that Charlie or we as neuroscientists are interested in. One method to investigate this is SSSI VASO, which has been developed for 7 Tesla by Renzo Huber. Here we use an inversion recovery sequence where we have a magnetization preparation period in the beginning, and then we acquire 2 images. One at the so-called blot nulling time, which basically gives us the CBV weighting, and then later a T2 star weighted image, which gives us a bold contrast. And here you can already see one challenge for these type of acquisitions, that we have this magnetization preparation in the beginning and we acquire 2 images. We're just inherently slower than with other types of sequences. The second problem is that these type of sequences are inherently SNR starved. So that is why most people employ these relatively conservative block designs with long stimulation durations and then long rests in between, which for neuroscientific applications feels rather rigid. And Charlie dreams about the flexibility to use event-related designs that are often used by neuroscientists to, for example, look at surprise effects or to disentangle the different the neurovascular coupling mechanisms. So to summarize our 2 problems, first, we have to acquire our images relatively fast. And secondly, we have to have enough detection sensitivity to actually capture short responses or responses to short stimuli. So to tackle these problems, we first designed a sequence for ultra-high field, so at 70. It is a SSSI VASO sequence with a 3D readout, and we managed to get our volume acquisition time down to below a second. Also importantly, we also kept a sub-millimeter resolution at 0.88 millimeter isotropic, and we acquired 16 slices. And on the left, you can see our coverage, and it was nice that we can both in our setup image the calcarein sulcus and the somatomotor area, which are 2 regions that are often used for depth-dependent imaging. If you are interested in the sequence PDF or analysis code, you can scan the QR code here, and it will give you my GitHub repository where everything is collected. We then tested first 2 participants in a visual tactile paradigm with flickering checkerboards and these piezoelectric devices which stimulate your fingers and therefore somatosensory cortex. And we first used the standard block design with a 30 seconds on-off, and we acquired 2 runs with our new short sequence and 2 runs with a longer, more conservative sequence to see whether we can actually use these fast-paced acquisition methods. And here, the important thing that we found is that in our visual ROI delineated on the bottom, across cortical depth, we see qualitatively similar results for the short and the long acquisition, which led us to conclude for both BOLT and VASO, and which led us to conclude that we can actually acquire images fast. To tackle the second problem with the detection sensitivity, we then acquired data from 10 participants, both again, all again with these visual tactile paradigms, but then we acquired run-wise, block-wise stimulation runs and event-wise stimulation runs, in which we stimulated for 2 seconds with a 3 to 10 second intertrial interval, which was heavily jittered. When we then look first at the responses to the blocks, which is our ground truth, so to say, or comparison, we can see in our statistical maps, this is from one subject, 12 minutes worth of data, that the maps look beautiful with the qualities of bold and vaso that we want. Namely for vaso, that we are centered in gray matter, whereas bold is more biased towards the surface. If we then look at the temporal evolution of the response, we see the nice sustained response with maybe something that we can interpret as an offset response and a larger undershoot for bold than for vaso after stimulus cessation. Across cortical depth, we also see patterns that we would expect. We have white matter on the left and CSF on the right, with peaks in middle layers for vaso and the draining vein, or the peak at the surface for bold, which exemplifies the draining vein effect that we talked about before. Now onto the events. This is on the left now activation maps for the same subjects, but now 2 runs, so 24 minutes worth of data. And we can still appreciate that we can capture these responses here estimated with a GLM. Of course, with a little bit lower signal intensities, but we still get something that looks very reasonable. On the right, we then see a deconvoluted signal here with a finite impulse response model, which shows a nice hemodynamic response for both bold and vaso, where then you could maybe even interpret the post-stimulus undershoot, which is more similar here for those short stimuli. For the layer profiles here for the events, we see a little bit more variability in the peak location for vaso. However, it is still far more biased towards the middle cortical depth as we would expect than bold. Of course, if we want to employ these designs in a flexible manner and also in an efficient manner, we have to see that we can get enough signal within a given number of average trials. Because if we would have to average hours upon hours of data, these studies wouldn't be really feasible in our modern context. So here I cumulatively averaged more and more trials together, and in one subject we can see how the response stabilizes. This is the dark hue, and in the light hue I plotted the response across all subjects. And we can see that after about 25 trials, we actually get a reasonable estimate of the overall response. To make this a little bit more objective, we then did this across all runs of all subjects. And we can see that, of course, again, we have a lot of variability in the beginning if we have a few averages only, but as soon as we average more trials, the response actually stabilizes relatively quickly. And as soon as this variance is about 5%, we said, okay, that is a reasonable estimate of signal stability, and this is for vaso at about 20 trials, which of course then depends on your stimulus, but for our strong visual stimuli, we thought that is quite reasonable, and yeah, we were happy to see that. So this led us to conclude that we can actually not only tackle the first problem, but also that we do have enough SNR and detection sensitivity to detect responses to our events, even with our tiny voxels and at the sub-millimeter range. One thing what was also very exciting is that we could look at these subtle laminar timing differences that vaso showed us. Here seen in a slower onset and peak time in superficial layers compared to middle and deep layers. We do not see this in our bold data. However, this has also been shown by other people, for example, Gironciero, so definitely check out their work as well. Finally, about the flexibility, we have data with somatosensory stimulation as well. So we also looked at somatosensory cortex. And here we get also nice maps and hemodynamic responses similar to the ones we detected in visual cortex, which led us to conclude that, yes, we can even use these stimuli in multiple paradigms and not only in the testbed of the visual cortex. So to conclude, my takeaway messages. Yes, we can use high field fMRI at 70 with sub-millimeter resolution to use these fast-paced acquisitions with vaso. we can capture these event-related responses within a reasonable amount of trials. And we are flexible in the choice of sensory modalities, and it can even be used to study neurovascular coupling. And yeah, if you are now like Charlie and think this sounds cool, or you maybe have any questions, you can either, of course, ask them now or come to the poster presentation on Wednesday and Thursday, WTH 1058. And with that, thank you very much, and I'm happy to hear your questions.
Well, now this talk is open for question. If there are any question, you can either raise your hand or use the app.
Okay,
So,
I actually have one question just to start things off.
Sure.
That's really nice. I noticed a couple of things with your transients. First, that, I mean, it looks like is there any chance that the latency is misestimated just because your baseline might be uncalibrated or something? And also, it looked like when you showed the whole time course before that it was almost showing a pre-undershoot of some sort.
Yes.
Could that influence?
So you're talking about this slide first?
Not even that one. A little bit later when you showed how you averaged them together, like
yeah.
averages.
Sure. So this one, yes.
Yeah.
So this is because we have a relatively fast-paced design, and these are computed as event-related averages, as you would do in regular EEG analyses. So I just epoch the data and average them together. So if we present our stimuli within consecutively like 3 to 10 seconds, we still have effects from previous or following trials, which will estimate influence this. Which should be less pronounced here, which is then estimated with a finite impulse response, where we do not see this effect.
Okay.
Yeah.
So any other question from the floor? Well, maybe I can ask you a question. Then in event-related standard fMRI, people just reported if you stimulated multiple times with short gap, then signal just has a linear property, right? It's just no summation occurs. I'm just wondering, in the case of event-related basal, do you expect that there is a linearity when you are just stimulating multiple times or stimulus getting longer? Do you have any expectations?
Sure, that is a very interesting question, and I would have liked to also investigate this experimentally to just vary also the intertrial intervals and see how that develops. We chose 2-second stimuli for just convenience and because we wanted to see how it works. But of course, yes, that would be a nice exploration, actually, yeah.
Okay. Any others?
Any more questions? All right, really nice work.
Thank you.
Okay, the next speaker is Francesca Medino, and her talk will be longitudinal and simultaneous whole brain fMRI and cortex-wide calcium imaging in awake mice.
Thank you very much. Hi, everyone. Okay. Yeah? Okay. So we all know that we can study brain function across a lot of different perspectives. And for example, if we take the spatial scale of events, sorry, mask off. Okay. We can study how the whole brain works, or we can focus on specific brain regions. We can go further than that in the spatial resolution and go like on neuronal circuits of events, and even further than that. The same happens with the temporal scale of events. So we can investigate more global signals, like the bold signal from fMRI, which is more indirect measure of brain activity, or we can go further in the temporal resolution and check how single cell recordings behave. And we can also interpret the temporal scale of events in terms of how the brain develops over time. Ultimately, the goal of one of the major goals of both preclinical and clinical research is to be able to better understand the human brain. This is why it's important from the preclinical standpoint to be able to better translate our results toward other species and eventually to the human. And that's why it's crucial now to be able to apply multimodal approaches that allow to overcome limitations of techniques that are otherwise limitations of the techniques, if they're used alone. So in this talk, I'm going to give a couple of examples of how we try to optimize our results and better translate them to the clinic. The first part is about a dual imaging setup, and then how we can keep animals awake in the scanner. So for the dual imaging setup, spoiler, we are talking about mice here. So for the dual imaging setup, it's a combination between fMRI and wide field calcium imaging. fMRI is going to be our common denominator to the clinic. So it's a non-invasive methodology that allows to have whole brain access. However, it provides non-direct information of the neuronal activity, and it's non-cellular specific when used alone. This is why on the mouse we can go a little deeper in the resolutions, and we can combine fMRI with wide field calcium imaging, which allows us to have more direct information on neuronal activity, especially because we can label one specific cellular population and extract that signal only. Of course, this methodology is more invasive and therefore is only applicable on animals. Okay, so in order to do our dual imaging system, we need a glowing brain. So we need a brain that expresses a calcium indicator for cell activity. So what we do is we use a novel methodology to introduce this calcium indicator, this fluorophore, in the brain by using a methodology called transverse sinus injections. So in this case, we inject in P0 pups, so the pups are just born. We inject in the transverse sinuses a virus that carries the fluorophore that we want. In this case, it's the GCAMP fluorophore. And because the BBB at this stage is completely immature, by injecting in the blood vessels, then the virus can travel through the entire brain, and we reach full cortical expression of GCAMP by P6. So after we do the injection at P6, the animals grow to be adults, and then at 3 months of age, we expose the skull to be able to do calcium recordings. So what we do is something called skull thinning. So we manually drill on the skull, and we make it optically transparent and smooth, however, still completely intact, and that's crucial for the MRI side of the story. Then we can, by adding glue and dental cement, we can glue a optical implant on top of the head of the mouse, and that gives us permanent access to the cortex. And this implant also doubles as a stabilizer for the head, because we can immobilize the animals through the same implant. This is how the calcium imaging looks like from above. And we acquired that by applying a fiberscope on top of the head of the mouse with the implant. The fiberscope is essentially a fiber optic bundle with some lenses and mirrors that allows us to deliver light onto the GCAMP positive neurons, and then we can record the activity of those neurons back in the same fiberscope. All of this, of course, is done inside the fMRI scanner. So that means that we can have simultaneous access of whole brain 3D fMRI data on one hand, and 2D calcium imaging data on the other hand. So now the problem is, how do we merge these two very different types of data onto the same space? So what we do is, from the MRI side of the story, we acquire an angiogram. So with the angiogram, we can follow the blood vessels on top of the angiogram and the calcium optical data, and then we can overlap the two and transfer all the calcium imaging data on top of the MRI space. So here I just show you that through our implants and the dual imaging system, we were able to acquire data for over 9 months of lifespan of the animals. So we implanted them at 3 months, and we imaged them at 4, 6, 9, and 12 months. And I was quite pleased to see that the implants held very well, and no implants got detached in this process. And we had stable calcium and fMRI data across all sessions. So with the dual imaging system, essentially we can leverage the fact that fMRI is non-invasive, therefore is applicable to the clinic. And with the calcium side, we can extrapolate cell-specific information that then we can extract from the bold signal of fMRI. And of course, with our implants, we can image the animals longitudinally, so we can apply this methodology on healthy and disease conditions. Okay, so that's for the dual imaging system. Now, awake imaging. So why do we want to do awake imaging in the animals? Most of fMRI, preclinical fMRI in rodents is done under anesthesia. And that's, of course, because it's really hard to convince the animals to be still in the scanner. And motion and stress are two major components that we want to avoid in imaging animals. So that's why we image them while they're sleeping. However, anesthesia, of course, affects the brain in a lot of different ways that we don't even fully understand yet. And most of human fMRI is done in awake conditions. And if we want to translate our results going towards the clinic, it's important to try to develop methods to image the animals while awake. And that's what we are trying to do here. And FYI, I didn't give coffee to the animals. It's just to show that they were awake in the scanner. OK, so first we did, over the 12 months of lifespan of the animals, we did 2 imaging sessions while the animals were anesthetized lightly. Here, I just focus on the fMRI side and on the motion parameter, which was what we wanted to really establish with this protocol. So on the y-axis, it's how much each brain volume moves compared to the other brain volumes. And on the x-axis is over time. These are all the data plotted for all the animals. And the red line is the threshold above which we would discard that brain volume because we consider it to have high motion. So that's the anesthesia session. Everything is nice and low in motion. Then after this one, we started training the animals to be awake in the scanner, which means we gave a first progressive training of 14 days. Progressive training because we introduced different sources of stress, increasing in numbers and increasing in duration every day. So the first one is handling the animals for a few days, then restraining the head of the mouse. The mouse is sitting on a hairnet. If you want to, we can discuss later about this as well. Third stressor is the LED. So we apply the calcium imaging fiberscope on top of the head because when we acquire calcium imaging, there is a lot of flashing light. So it looks like a disco ball essentially inside the scanner. So we wanted to give that as a stressor as well to habituate them at the light. And finally, of course, the fMRI noise. So after this 14 days of training, we did an initial awake session, which of course led to more motion compared to the anesthesia session before. These are, again, all the data plotted together, so that's why it looks very crowded. I will show you later the quantification of this. But this is the motion for the awake one, so after the first progressive training. After this, we refreshed the memory of the animals for 2 days, where we provided all the stressors all at once with full force. And then, of course, second awake imaging session, which in this case led to a drastic reduction in motion. So we can see already that the effect of the refresher is quite prominent here. So if we quantify this, here I sort the motion from highest to lowest, and we can see that the blue line is the first awake session, the pink line is the second awake session after the refresher training, and we can definitely see that there is a reduction in motion of how much data we'll discard. So in the first case, we will discard 18% of the data, second case, 9%. Yeah, so of course, the refresher really works in this case. An interesting thing that we noticed is that I told you that this is the fMRI side of the story. So if we look at one specific run for one mouse, I chose this one because it has a very high peak in motion from the fMRI side. If we look at that specific peak in the calcium data, we see almost zero motion because the scale of the motion is basically null. So what we can assume and kind of conclude here is that motion in the fMRI is predominantly due to the z-axis, and we think it might be because of the jaw of the mouse moving. So to conclude, if we go back to the initial slide, with our dual imaging system, we have a way to overcome limitations of different techniques, and we can bridge the gap between different resolutions, both on the spatial and temporal scale. We can image animals across time and see how the brain develops in healthy and disease conditions. And because we use the transverse sinus injection method to inject the fluorophore, these viruses are commonly available and can be bought in like regular websites. So that means they can be adapted to other species as well. So from mice, we can go up to rats or monkeys even, and this way higher order mammals. And finally, by imaging the animals while awake, we can definitely bridge the gap of the anesthesia. Then, of course, we can discuss more about the implications of awake imaging in the animals. So if you want to talk more about this, I have a very sober poster that you can come and visit on Wednesday and Thursday. Or if I'm not at this one, I have another very sober poster that also you're welcome to visit. Thank you very much.
Well, now this talk is open to the question. Are there any questions from floor? Yes, one over there. Yes, please.
Great talk, thank you. My question is about the expression of the virus. I was just wondering why you choose the transsinus expression instead of just pick up the transgenic animals.
Yeah, that's a great question. So in our case, this is part of a bigger project where we work with a double knock-in model for Alzheimer's disease. And that model is really painful to breed and crossbreed with the GCAMP line. So that's why by using this methodology, we just take the pathology model and we inject them from day 0. This way we can have the expression of GCAMP without having like cycles of expensive and like a lot of mice. So that's how we try to avoid that.
We have time for one more? Okay.
Yes, please.
So in terms of motion, you said you lost 10 to 18% of the data, I think, depending on the situation. how is that distributed? Are there certain animals who don't acclimate well, or is it kind of evenly distributed across animals and runs? Have you looked at that?
That's a great question as well. So for now, we haven't looked at that too much in detail, but I can see that, for example, females seem to handle it better than males. But we use all, like I put all the data together here, like as an average of all mice, but we see that females handle it better. But again, there is a lot of, I think also like Mallar's group, they're looking at differences in stress in animals between females and males. So we definitely need to go down that route for sure.
Okay, thank you.
Well, maybe I can just pick a few questions in the app. Maybe there are two related questions. One is how do we make sure the animals don't suffer with skull drilling? Then second one is how do you differentiate that the effect of reduction of motion is the effect of habituation and not a fear response?
Can you repeat the first one, sorry?
How do you make sure the animals don't suffer with skull drilling? I don't know.
With what?
Well, maybe you can skip
Well,
those. Yeah.
I guess there's a concern of the initial operation of, taking off their skull.
Oh, skull drilling.
Yeah.
Yeah, so we basically, like, by looking at the surgery, we ensure that the animal, like, the skull is intact. And by applying the implant, those implants were put on at 3 months, and then they stayed on until 12. We didn't see, like, at the end, we could see some deterioration at the end, around the edge of the surgery, but not in the middle of the brain. So I would assume that there was one case, for example, where the skull was punctured, and there was a full regrowth of the blood vessels within the glass. That was quite cool, but I couldn't show it here. But yeah, so we would discard those kind of animals, but it was only one case in this one. And what was the second question? Sorry.
Well, how do you differentiate that the reduction of motion is an effect of habituation and not a fear response?
So if the animals are older, that means? Yeah, the only way to find out is to scan them earlier, I think. And that's exactly the next step, to habituate them earlier and scan them earlier.
Okay, I'll be looking forward to what you find with this as well. So, all right, thank you.
Thank you.
Let's move on. So next speaker is Yang Woo Park. The title of the talk is Super Resolution in Vivo Human MR Imaging.
Hello, my name is Yang Woo Park, and I'll be presenting our proof of concept work on super resolution in vivo human MR imaging. So high resolution MR imaging with resolutions of 100 micrometer per voxel or above could significantly enhance visualization of brain anatomy. And this could have clinical uses in detecting small lesions in diseases such as epilepsy or MS. And it could also help us understand the small and fine regions such as cerebellum. However, any increases in spatial resolution comes at a cost to signal-to-noise ratio, and that is because MR signal comes from protons within a voxel, and having a higher resolution image means signal will be coming from a smaller volume because of the physics. Now, one of the ways to rectify this is, or alleviate this, is to use a higher magnetic field scanners, such as 7 Tesla ultra-high field scanners. And theoretical possibility of protons is directly proportional to the field strength. So ultra-high field will have better SNR. But then the gain alone would be insufficient in the increases in the smaller volume. As you can see from here, from 3 Tesla to 7 Tesla, you get 2.3 times the gain. But then if you have each axis, it's a 0.25 loss, so you're still going to lose signal. So you're going to be affected with the signal loss. You still have to deal with the poor signal to noise, and also as you increase the scan, as you increase the resolution, it lengthens the scan time because readout length is longer, and you may need to do some signal averaging because of the signal loss. And also higher MR field just changes the T1 and T2 coefficients. And also those familiar with the scans in high Tesla knows that there's, as you shorten the time or you change the parameters, you're going to hit with this safety SRR limits. So there's a lot of restrictions in acquiring higher spatial resolution images. So I was looking for solutions, and one of the solutions that I found was that I took inspiration from DSLR cameras. DSLR cameras, especially in the old days when they have limited sensor sensitivity, was that they acquire multiple low resolution frames and combine them to acquire high resolution frame, and it's called pixel shift by certain manufacturers, but it's generally known as super resolution. So, in this example, you see a low resolution image shown here, and then, as you combine these frames, you see these, sort of details. So I thought, maybe I should take this super resolution and try to generate really high resolution image close to 100 micrometers per voxel. Now, so just to explain what it is, like I said, it's to combine multiple low resolution frames and to generate a single high resolution frame. And here the orange balls represent individual pixels in an image. And these are basically the probability density function of protons in that location. You can think of it that way. And then if you acquire another with the physical shift, like you do with the pixel shift, that has its own probability density function. So you use those information to generate these intermediary frames. So it's sort of, it's sort of a little, I suppose, a little better than interpolation, but so in between like averaging and interpolation. And just to note, these technologies had already been used in MRI before, about 10 years ago, but it's mostly used for scan time reduction in compressed sensing. So I kind of wanted to use this for pushing the spatial resolution. So for this scan, for this proof of concept study, acquired some data from 7 Tesla, and I try to use commercial hardware. So I use the 7 Tesla Siemens commercial scanner using the commercial single transmit coil from Nova Medical. I try to stick with whatever the Siemens provided, but I had to acquire or had to modify the sequence a little bit to increase the limit because it's physically limited to 1024, and you could, unlock to 2048, but then there's a lot of restrictions, so you can't really, it's difficult to push all that. But yeah, so I had to change this bit, but otherwise it's, standard sequence from Siemens. Now I use, you need to do sub-voxel shift, and I use this tool called AutoAlign, which comes with Siemens scanners, and try to control sub-voxel shift using this. And then I use the publicly available super resolution code. I mean, there are a bunch of codes around available, so you can use those use one of those MATLAB-based code to generate the high resolution images. Just to point out this AutoAlign, so it's a tool to position where you acquire voxels in Siemens scanners. And you can sort of override where the image center is, and you can sort of control the offset in the protocol editor. So for this one, I acquired 4 frames, and I each set I had a shift in either left or right or anterior posterior directions, and AutoAlign will automatically compute where you're going to acquire. So there's physical shift in the acquisition images. So wanted to test 3 different questions that I had. Where first one, how is it going to actually compare against the high resolution? So I acquired a high resolution image, and I have the resolution, and then I acquire 4 different frames and try to compare the two images. I also wanted to see because mostly the high resolution images are T2 at the moment because of a lot of constraints. So I just wanted to see if it's going to apply for other types of images. So I acquired T1 and SWI as well, and try to get what's the maximum sort of resolution I could acquire. And the last thing was, so these are, the first two are the 2D slices, so you have really thick slices. But I was wondering if I could acquire 3D volume with isotropic voxel dimensions. So one left is a higher resolution image I acquired, and these are the parameters. And I have a poster that has all these parameters as well. And the right side is a super resolution, and it's comparable. So if you zoom in, the high resolution, it has more detail. It's, the edges are sharper, but then there's some streaking artifacts because each block is a lot longer than with the super resolution. You have shorter blocks that you acquire 4 times, whereas this one you have longer block. You acquire 2 times because of averaging. So, and then the tissue, I was surprised to see the white matter and gray matter. The separation is better displayed in super resolution versus high resolution. So, I think the bottom line is it's comparable. It's hard to say one is better than the other, but it was interesting to see super resolution, comparable images with super resolution. And then I acquired with different contrast, and it's just the same thing. You acquire 1, and then you just acquire 4 frames. So with the T2, I try to push the resolution all the way. So I acquired at 0.21 by 0.21, and then did SR to 0.1. So and with really high resolution, you start to see a lot of details, which was interesting. And each one is about 7 minutes. So with 4 sets total, it's about a little less than half an hour. So it is still a long time because you just the high resolution, you just need longer time. With SWI, it was interesting. I tried to acquire from same location, but then the sequence is a little different. So, but yeah, you do see a lot more details with the high resolution. And T1, it's the one that I had a lot of issues. And that is because with T1, you need to have short TR and short TE. Short TE, there's a problem because the readout length becomes long. So if you try to increase the resolution, you're just going to have longer TR or TE. TR is difficult because, with the SAR, you just can't really shorten the TR. So T1, I struggled, and at the end, I just used the MP range, which I acquired at 0.6 by 0.6 by 0.6. It just did SR to 0.3 by 0.3. So it's one of the areas that I think needs a lot of work in the future, the high resolution T1s that doesn't use contrast agents, I want to point out. And then lastly, I wanted to acquire the 3D volume. So I acquired chrono slices at 0.3 by 0.3 by 1.2. And because 1.2, you can divide by 0.3 and it's 4. So I acquired 4 sets. So each one, for this one's longer, so it's about 11 minutes, but I acquired 4 sets. So you can see from here, each one's kind of squeezed because, the anterior posterior dimension is 1.2 millimeter. But then when I did the SR, you get you sort of generate this sort of flat image, and then looking at 3D, you see sort of a volume, and so within 40 minutes you can acquire this, and I didn't use any motion trackers, so I was happy with it. But then there's one issue, so I wanted to look at cerebellum, but then currently with single transmit coil, you lose a lot of signal, so you don't see much cerebellum other than the edges, and this was after using dielectric padding, so there's still a lot of work to do, so in this regards. So the bottom line, I have shown that you can generate really high resolution images with super resolution, and there's advantages and disadvantages, but I feel it's comparable, and it's something that we could explore more. I feel there are a lot of talented engineers and scientists here that could tackle this. And then, it's sort of contrast agnostic, so you can try with different contrast. But there are still a lot of challenges that need to be solved to acquire different contrasts at really high resolution. And then I was able to show the 3D volume. So hopefully something like this could be used in addition to a lot of volumetry analysis, and that can help understand different regions in the brain. And I just want to finish off with this slide. So, I took the inspiration from how the DSLR cameras are acquiring brain images, and I think we could also do the same. So, we can take inspiration from other modalities to solve these challenges with MR. So one of the things that I've noticed, and it's apparent, is that deep learning has been used extensively in other domains. So with the machine learning, we could do super resolution, and I think there's some abstracts that does mention this. So we can try to push the spatial resolution with machine learning. And another thing I want to point out is that so in phone cameras, they use this technique called image fusion. So you acquire different types of images, but you acquire really fast, and you acquire multiple frames, and you combine that. So it's kind of like MR fingerprinting, but we can sort of think, we don't have to be fixated with one contrast. We could acquire multiple contrasts and then sort of synthesize. I don't know, it kind of sounds bad, but we can sort of generate and get really high spatial information from that. So yeah, I'm just throwing these ideas. Thank you for the collaborators that helped me out with this, and then I would like to thank the organizers for giving me the opportunity to present our work, and also funding sources. So thank you very much.
We have a lot of questions coming up. There was one related to case space, but before I ask that, the first question that always pops into my head is when I've seen this before, and this is really nice. Actually, it's nice and straightforward. It has a shorter readout window, and it might be applied to echoplanar in that regard too. But you could pretty closely do a similar thing, just keeping everything in K-space, and just doing segmented K-space. Have you considered trying to compare those in some way?
Yeah, I think, obviously I just wanted to see if it works. So there's a lot of, I need to develop this work a little further. I haven't done any serious signal to noise or anything, but yeah, there are a lot of literatures that does this in case space, and you sort of try to increase the resolution that way. So that's one of the next steps that I will be looking at. Yeah.
Yeah, because I see potential advantages of this over that, or it's interesting. The comparisons are not obvious. But
any question from the floor? Yes. Yeah, please.
Yeah, it's actually a continuation from Peter's point. In K-space, when you have a shift of half of pixels, it's really just a phase change in K-space. So how different is it to acquire this in this way, or just manipulating effectively the K-space and doing multiple averages? I mean, how much more information you get? And this is something I always get a bit of a problem with this super resolution in MRI, because you acquire your signal originally in K-space, not in image space. Yeah, so that's one of the reasons why I decided to stick also with the simplicity. I was just doing this with DICOMs, but yeah, and I was aware of these issues with doing all these in K-space, and there are papers that refutes this approach. So I'm aware of it, and it's something that needs to be solved. But I just wanted to test out, and it was working. So, but there's a lot of work that needs to be done. But I'm just saying, with 7 Tesla or 10.5 or whatever you have, we're hitting this limits, with the hardware, and just going high Tesla
just doesn't seem to cut it. So
Yeah. Yeah, the one potential advantage I see of this is that, like, in K-space, right, if you collect at different times, there's different amplitude modulation in K-space that you have to correct for, and here you don't. But yeah, it still seems that keeping it in K-space is ultimately better if you can correct for that. But still, this is nice and straightforward and practical. And
And I think that's one of the reasons why I put the last slide, because getting multiple frames, you can think about getting like maybe low resolution contrast, and then high resolution, like the outer edges of the K space. So, it's something that I'm thinking about. But,
So time is up. We have to move on. Thank you very much. Okay. The next speaker of the session is Sara Sanchez Alonso. The title of the talk is Within Subject Correspondence of Simultaneously Corrected fMRI and Full-Head NIRS Signals.
Thank you. Thank you. So I'm going to present today a study that we've been collecting, that we've been running the last year. And it's a study in which we are collecting simultaneously fMRI and NIRS with full head coverage. First of all, I'd like to acknowledge my collaborators, Adi Kaslin, Rebecca Canal, and Isabel Nicholson. NEREX is providing the NIRS device that we are using. And also, I'd like to acknowledge the funding sources, the Bill and Melinda Gates Foundation and NIH. So what's the motivation for the study? Why do we want to compare these two different signals? So for those of us interested in studying neurobehavioral variation across development, one of the challenges of fMRI, especially when working with infants and toddlers, is that fMRI, although is the gold standard methodology, in adults, it is very difficult to implement with infants, with toddlers, even young children, especially awake fMRI. In addition to that, we need methodologies that allow studying the infant brain outside the scanner, right? If we're interested in studying language development, social development, those are key. The environment is key in early development. So what we've been doing is we've been conducting a quantitative comparison of fMRI and NIRS signals across development. Two of the key features is not only that they are collected simultaneously, but also that this is the first study that is using NIRS with full head coverage. We are doing this with adults and children. And really the question that we are asking is, to what extent are the two signals comparable across development? So they both rely on the same neurovascular response, but we don't really know to what extent can we compare them, right? And today, I'm going to just focus on the study that we've been running with adults, given that it's the most complete that we have at the moment. I'm going to present an overview of the main challenges of running this kind of a study and some of the preliminary data that we have. So NIRS, as many of you probably know, is particularly advantageous for studying development, especially early development, because it can be used in naturalistic environments. There is no noise. It's a portable methodology. And I'm just going to mention a few other key properties of NIRS in comparison to fMRI. In addition to allow us to test in naturalistic environments, it's wearable. It's just a cap that the child wears. It's portable, so it can be moved to different places. It's much more robust to motion than fMRI, and it is much less costly as well than fMRI. So those are some key properties that make it very attractive for developmental work, especially the first 5 years of life. So I'm going to talk a little bit about the equipment that we are using. So we are using a NIRS Scout system. Here you can see a picture of the cup that we are using. The optos, they have flat surfaces. This is so that the subject can lie on the cup in the scanner. We are using a high-density full-head cup with 105 channels. The optical fibers are quite long. As you can see here, they are 10 meters long. And the way that it works is that we have the equipment, the NIRS equipment outside in the control room, and we bring the optical fibers into the scanner room through the waveguides. And here you can see how we have to design this bridge that you can see here so that we can bring the optical fibers into the scanner table. As I mentioned, this is the first time that this is done, so we really have to try different things and try to figure out what actually worked best. And this seemed to be the best solution for us so far. And so this is how the system looks in the scanner. For data collection of the fMRI data, we are using a Siemens 3T Prisma. We are using the 20-channel head coil because the 64 and the 32 didn't allow us to, the NIRS cap didn't fit with ADO, so we have to use the 20-channel head coil. At the same time, we are simultaneously collecting physiological measures using heart rate and respiration. We are collecting in particular heart rate and respiration rate, as you can see here. And here's a picture of the NIRS array that we are using. As you can see in this image, we are using short channel measurements. These are what you can see here in the locations are in green, picture on the array. And this is key for our study because one of the main sources of noise in the NIRS signal is noise coming from extracerebral layers, so coming from the superficial layers from the skull. So basically the short channels, what happens is that the detector and the source are located at 8 millimeters, such that any signal that we collect is just extracerebral signal, and we can regress it out from the NIR signal that we are collecting at the same time with the long channels. You can see here at the bottom right how the short channels look. Another important thing that we need to do is to be able to localize the optot on the anatomical images. We are using two different methods. Since we have the T1 images, we are localizing the optos onto the anatomical images of the subject. So we are using for that vitamin E capsules as fiducia. So the vitamin E capsules are located on every optot of the NIRS cap. You can see here in this image that they light up in the scanner, so they are easily identifiable. In addition to that, we are using a 3D structure sensor. This is the sensor that we usually use outside of the scanner when we don't have a T1 image available. And that was important for us. It's important for us to do this comparison because that's going to allow us to see what is the margin of error between using a T1 or using the structure sensor. And I'm going to talk a little bit about the structure sensor for those of you not familiar with this device. So basically, the way that it works is that this sensor projects what is an unknown pattern, in our case, the head, onto a scene. And the way the pattern deforms when striking the surfaces allows the scanner to calculate the depth and the surface information of the object. So as you can see in the picture, what we do is that we locate some key skull landmarks, and then we just go around with the sensor, creating this 3D surface of the head that later on we are using for optolocalization. So this is how one of the subject's mesh looks after we've done the scanner with the 3D sensor. What we do afterwards is that we mark the fiducials on the subject's mesh. We could register the 3D scan with our coordinate system. This seems to be more accurate than using just 10-20 models and results in better forward model solutions. So we are using this system just as a comparison with the T1 because that's the system that we usually have available outside the scanner. So just a little bit about the experimental design that we are using. So we've collected data from 40 young adults. We just finished data collection actually last month. And the subjects, they come for 2 sessions. In one of the sessions, they participate in 2 movie watching sessions for a total of 15 minutes, and 2 rest and stay sessions for a total of 10 minutes. There is a second session that they also do. They do another 2 rest and stay sessions. This is to assess the retest reliability of the signal. They also perform a traditional language task using a story versus math contrast and a visual task using a flashing checkerboard paradigm. You can see here just an overview of the pre-processing and analysis pipelines that we are using. I just want to highlight a few things since I don't have time to go in detail on that. So for the fMRI data, we are using standard HCP pipelines. For the NIAS data, we are analyzing both the light level time series. We are using and we are also doing image reconstruction, which means basically that we are co-registering the optos onto the anatomical images, and in addition to that, we generate sensitivity profiles of source detector pairs. This is important because the levels of scattering and absorption of the infrared light is going to vary depending on the underlying brain tissue. So that's something essential for comparison of the two signals. And then we map both signals onto the cortical surface, and that's at the level at which the comparison happens. So I'm going to just present some of the preliminary data that we have from the visual task. This is the flashing checkerboard paradigm. And for the checkerboard versus rest contrast, here I'm showing the NIAS data. It's just within subjects. It's just individual level data. And what we can see here is a couple of things. We see that across subjects at the individual level, we see engagement of primary and secondary visual areas. As you can see with the NIAS data, we can collect both oxygenated and deoxygenated hemoglobin. And as you can see in the maps, the two signals are negatively correlated, which is what we would expect. With respect to the fMRI data, there are no surprises here. I'm just showing the beta maps just to show that there is as expected engagement of primary and secondary visual cortex. And if we look at the subcortical level, we also see engagement of the LGM. So I don't have time to go into more detail into any of the data that we have, but I'm going to do a summary of what I've just presented. So our study builds on prior work, and it innovates in several ways. It uses full head coverage for the NIAS data collection. We are measuring simultaneously physiological signals. As I mentioned, we compare two co-registration methods, and we are also assessing retest reliability of the signal across two different sessions. As I've shown, subject-level results show modulations of visual cortex areas across both fMRI and NIAS. And our pre-processing pipelines include, crucially, regression of the physiological signal using short distance channel measurements, and also simultaneously collected physiological signals. And another important feature, as I mentioned, is that the correspondence of the two signals is made via comparison of two different co-registration methods. One of them is using the T1 image, the other one is using the 3D structure sensor. And if you'd like to know more about the study, please come to my poster on Wednesday and Thursday. And I'll be happy to answer any questions.
So now talk is open to the question. Are there any questions from the floor? Well, I can just start with a question related to somebody's commented in app. So if you have the fMRI, maybe you can just get signal from LGN, somewhere very deep area, but maybe functional NIRS can just get signal from more superficial brain area. So I think if you're just registering these two data sets, how do you just account for these differences? Because maybe some deep area, maybe you couldn't reach the signal. How do you just compare these two signals, which have very different sensitivity for deep areas?
Right, that's a very good question, and that's one of the limitations of NIRS, right? It doesn't allow collecting data at the subcortical level, right? There are different ways in which we've thought of doing that. One of them is only using the cortical data, the cortical level data of fMRI. That would be one way of doing it. Another way that we are also doing, we are collaborating with a different team at WashU, in which we are doing a simultaneous NIRS from fMRI. So that allows us to see to what extent can we use the fMRI data to create NIRS data, and using only the signal from the cortex, right? So it's a great question. It's very challenging, right? There are different ways in which we are trying to tackle the question, but yeah, we'll have to see what we find.
I actually have a question. I mean, this is great work, and it could probably shed a lot of light not only on development and neurovascular coupling during development, but even on fMRI on BOLD. I noticed, and I always sort of see this in past data as well, is that the ratio of deoxy to oxy that you showed, it seems like it varies spatially.
Right.
And that's always intriguing if there's like a blood volume effect that's different in different areas or something. I don't know if you'd want to talk about that a little bit.
Sure. Yeah. So that's something that is well known, that there is a special differentiation with respect to the ratio of oxy and deoxy. I don't know of any study that has done a systematic investigation of why, to what extent, but it is known that that is the case. Yeah.
Yeah. I mean, it could be interesting to look
yeah,
into.
for sure. Yeah.
Any other questions? Yeah, please. Here's a microphone.
I was just wondering, for putting these two data sets together that have such different coverage, what do you plan? Are you kind of calibrating FNIRS with fMRI to show that FNIRS can be used separately, or can you put them together in a single way? Are you looking for a spatial overlap of something, or are you trying to kind of spin off FNIRS based on fMRI, or are you trying to inform your fMRI knowledge more from the FNIRS extra data? It's just very hard to combine very different modalities like this. So what does that part look like?
Right. Yeah. So I'll tell you, how it started, and then, other things that we've been thinking along the way. So in principle, the two signals rely on the same type of neurovascular response, right? Increasing oxygenation as a result of neuronal activation, right? So the signal that we should be capturing should be very similar, in a way, right? And we know that from other studies. We know that other studies that have focused on ROI or motor function, we know that that is the case, right? In our case, our main goal would be to be able to, say, collect NIRS data in early development, and compare that to fMRI either later in life within adults, or at the same time, let's say that we have resting state collected during fMRI, right? So our goal is to use them separately, right? And with this study, what we are trying to see is, to what extent can we say that the signal that we are collecting with NIRS is also the one that we are capturing with fMRI, and the variation across development crucially, because prior studies have shown that the two signals correlate quite strongly in adults, but nobody really has looked at young children or early development, right? So that's still a question. Yeah.
Okay. Well, thank you very much.
Yeah, thank you.
So our next speaker is Gabrielle Ramos Lorden, and she will be talking about distortion and ghosting-free high-resolution ex vivo human brain diffusion MRI up to 50,000 seconds per millimeter squared. And I think that's she couldn't be
this
here,
is yes, video presentation.
Hello, thank you for coming to my talk, Distortion and Ghosting-Free High-Resolution Ex Vivo Human Brain Diffusion MRI up to p-values of 50,000. I am Gabriel Ramos Jordan, and before going to this project in detail, I would like to situate this oral presentation in the broader picture of the Connect on 2 project, a project that we are developing at the Martino Center, where we are developing the next generation of human MRI scanners to bridging studies of the macro, meso, and micro connectome. On top of developing the next generation of gradient system, we are interested in doing the multi-scale validation with diffusion MRI, micro-CT, and electron microscopy. And in this validation, ex vivo diffusion MRI plays a very important role because it's ideal for mapping the mesoscale structure of the human brain with unprecedented detail. And this is because we can achieve much higher spatial resolution than in vivo diffusion MRI, because we don't have time constraints, we don't have motion, and we can use a specialized pool sequence that renders high SNR, like 3D multi-shot EPI. all of the information derived with ex vivo diffusion MRI can be translated to in vivo diffusion MRI. So this technique is key. It's a key bridging technology for understanding, for mapping the human brain. Nevertheless, there are some peculiarities with ex vivo diffusion MRI. One is the fact that because the diffusivity of the tissue is reduced, to provide enough diffusion contrast, we have to use much higher p-values than normally we used with in vivo acquisitions. So if we want to have high SNR, we have to get a short acquisition time, which the only option is to use gradient systems with ultra high gradient strength, like the connectome system. One of the problems with these powerful scanners is that the strong gradients create eddy currents that affect the image readout, and that introduce perturbation, phase perturbation in the k-space encoding, which is not, if it is not properly accounted for, create ghosting, extremely ghosting, and distortions, which invalidates all of our analysis of diffusion MRI. Even conventional techniques like linear phase correction that is included in any Siemens scanner are insufficient here because they do simplistic assumptions of the phase variation between odd and even in EPI, which definitely do not hold when we use ultra high gradient strength. So clearly for ex vivo diffusion MRI, we need superior techniques. We need more advanced reconstruction approaches. One of those approaches is spatiotemporal field monitoring. The idea is that with a field camera, we can measure the spatiotemporal variation of the main magnetic field, and that gives you complete information of all of the perturbation phase as well as the k-space trajectory. We have a field camera in our scanner where we fit the phase, the measure of the phase, to a basis of solid spherical harmonics, where the first order represents the k-space trajectory, the third order indicates the V0 term, and the second orders and third order terms are unwanted components that we would like to be minimized. But as you can see here, we have observed that second order and third order terms are substantially higher, and this creates a strong ghosting that cannot correct with the linear phase correction. Nevertheless, because we are lucky and we can measure the spatiotemporal field variation completely, we can change all of our framework for reconstruction and develop a reconstruction techniques that are phase informed, and they can incorporate all of these variations in a model-based approach. Indeed, because we have the phase, the measure of the phase, we can fit the data to a spherical harmonics. We can create what is called the encoding matrix, which is a complex exponential where the phase is given by this equation. We have the co-sensitivities estimated from a gradient we call echo sequence that we can acquire prior to the scan. If then we have the acquired k-space data and X is our ghosting free, we can formulate this linear least square problem with Tikhonov regularization and solve for X, where because we have been including the phase variation in the model, we can achieve ghosting free images by solving this very easily reconstruction approach. It can be shown from these results that ghosting is indeed well reduced when model-based reconstruction with high order terms is implemented, as you can see here on the right, compared to the conventional reconstruction of Siemens. And we have demonstrated that this improvement is consistent over the whole brain. See here axial, sagittal, and coronal slice of a 3D multi-shot EPI dataset acquired at 0.8 millimeter for p-values of 4,000. Note that the ghosting is clearly reduced compared to the linear phase correction approach. And of course, this has an impact on diffusion MRI analysis. If we don't do any correction at all, not only we have visually unpleasant images, but also we are biasing all of the connectivity measures. We are biasing the phase, as we can see in the red square that I am pointing out here. We are biasing the orientation of the primary eigenvector of the tensor. We are biasing any tractography results. As a result, we are biasing the FA, and of course, we also suffering from unwanted artifacts in the mean diffusivity, which can be well corrected with our technique because we are incorporating high order terms in the image reconstruction. Another benefit, before going to another benefit, also this of course has an effect in more advanced diffusion models like DKI. And another benefit that I would like to address, that I mentioned in the title of this talk, was the fact that we can achieve distortion free. Of course, we know that there are very popular techniques like eddy from FSL tool that can correct for eddy current distortions by doing a registration. Nevertheless, this registration is not always perfect, especially if we go to super high B values like 50,000. And also, even in the case that the registration it's accurate, it always leads to blurring in the reconstructed image. And this is because the image registration always needs an interpolation. And no matter if it's cubic, this is going to blur the image substantially. And maybe this is not a super important issue when we are dealing with low resolution diffusion MRI, but definitely when we go to high resolution diffusion MRI, like in ex vivo, and we want to observe or to measure microstructure in areas like the hippocampus, this is gonna have a huge effect. As we can see here on the image on the right, with our high order image reconstruction technique, this is not a problem because it's a one-step approach, and all of the information is incorporated in the reconstruction step. As a conclusion, we have demonstrated here that ex vivo diffusion MRI requires advanced case-based reconstruction to deal with ghosting, because the strong eddy currents created with ultra-high gradient strengths create phase modulation that are not well modeled with the conventional techniques. They fall short, and model-based reconstruction approaches, like the one we are presented here, where we measure the spatiotemporal variation of the main magnetic field, and we solve the reconstruction problem as a phase-informed inverse Fourier problem, leads to much better reconstructed image, ghosting free, that leads to improved downstream analysis, and also images with less distortion and less blurred with high resolution. We also expect that this technique can be translated to in vivo diffusion MRI. And this is the end of my talk today. I hope you have enjoyed the talk, and please don't hesitate to contact me if you have any questions.
So, well, you can just leave the question in the app or just directly reach out the presenter using email, please. Thank you.
Or I don't know how long they'll stay on the app, but maybe they'll look at it there. So.
Okay. So next one is a last talk, which is a neuroanatomy. So this is the last talk in this session from Jordan Decrecker. Then title of the talk is Fusion of 3D Histology and Polarized Light Imaging via Hippocampal Unfolding in the Human Brain.
Okay, thank you for being here. All right, so I'll just jump right into it. So first of all, what is PLI, or polarized light imaging? Basically, this is a microscopy technique where you take advantage of the polarity of light in order to detect coherent fibers in the brain. So this could be axons and things like that. You can also additionally look at the angle the light strikes this tissue in order to make some inference about out-of-plane tissue information. So in particular, in this talk, I'm going to focus on just one measure called transmittance. So basically, it's proportional to the sum across different polarity directions. As you can see in this example image, it will show cell bodies as and white matter are dark, and the gray matter is mostly bright here. So the data set that I'm working with consists of one almost full 3D stack of an entire 3D hippocampus, meaning that we can build a 3D model of this hippocampus at microscale resolution. And my overall goal is to fuse this with this other data set called 3D Big Brain. If you guys haven't heard of it, it's basically one full brain that's been histologically imaged, or I should say imaged microscopically, and then every single slice was put together and reconstructed, making, as far as I know, the most detailed model of the brain that we have. So it's stained for neuronal cell bodies. The work that I'm going to talk about was done at 40 micron isotropic. So previously we looked at the hippocampus of this data set, and we found lots of interesting features, including extensive folding or gyrification or digitation, if you like, within the hippocampus, and lots of other interesting features like this. However, that may have been a bit of a warning of a technical challenge to come. So as I said, we want to now take our PLI dataset, which is at the bottom in this image, and fuse it to the original 3D Big Brain, so our very high resolution full brain model. And what you might notice is that I've highlighted the gray matter of the hippocampus here, and the folding patterns, there's not necessarily the same number of folds, or they're not in the same position. So when we try and run this registration between these two data sets, I'll just toggle this back and forth. So I'm taking the label, the gray matter label from the bottom, and propagating it to the top, and you can see it doesn't look so good. And I believe the problem is that it's really in these folds. So it's very hard to run a 3D registration when the folding or the gyri between these samples doesn't align. So this is the problem that I've aimed to tackle. So just interim conclusions. What we want to do is build this really detailed model, multimodal model of the human hippocampus. However, we're really worried about this inter-subject variability, specifically in folding pattern or gyrification pattern, if you like. So the solution I'm going to propose is to perform a registration in unfolded space. So conceptually, this is going to be very similar to a neocortical surface-based registration. So first, I'll just tell you a little bit. This is some previous work where we developed a tool called HipUnfold, where basically we get a very high detail segmentation of the hippocampus first, similar to a neocortical ribbon, so just mainly showing hippocampal gray matter. And then we just break that gray matter down along a set of sort of geodesic coordinates going from anterior, posterior, as well as medial lateral, or sort of along the inward C-shaped curl of the hippocampus. So so that allows you to then use two coordinates to flat map this hippocampus onto an unfolded coordinate space. Okay, and I should also note that this is basically topologically constraining these hippocampal, and in the different colors here, I'm showing hippocampal subfields. So I'll just make note of that. We then applied this unfolding system to the big brain and the polarized light imaging segmentations, and we created these 3D models in folded and unfolded space, where the different colors here represent manually done subfields done, or well, denoted in the native space. So along with this unfolding, you can then map out a series of features about the hippocampus. So for example, I'm just showing the extent of gyrification within these two samples, the thickness of that tissue, and the mean curvature of that surface as well. And what we can then do is, if we want to improve the overlap between these two samples, we can potentially do a registration in this unfolded space based on these morphological features. So note that these are contrast agnostic features, which is what we did here. So we just used ANTS registration, so a common registration toolbox, and we generated a warp field that we then can apply from one subfield definition and transform it in the unfolded space to the other subject. And then we can also refold it, I suppose, in that subject's native space as well. So I'm just showing that both quantitatively and qualitatively here. So just looking at the images on the right, we have the original PLI 3D model, the big brain reference on the right, and some attempts that we had made to register these. So initially it was just a 3D native space registration, where you can see there's all kinds of issues arising because these folds are not well aligned to each other. And then in the middle, the unfolded topological alignment. So this is basically we just unfold one sample and then refold it in the shape of the other sample. And then the third one here is unfolded with registration done in this 2D unfolded space. So again, conceptually, this is similar to the sort of inflation to sphere and then surface registration we might do in the neocortex. Luckily, like quantitatively, we see quite a stark improvement in the overlap between these manually defined subfields, which is kind of our ground truth for how homologous are these two points between the two samples. Altogether, we think that doing this registration in unfolded space makes the most sense because of the sort of inherent 2D topology of the hippocampus, and it makes this a more constrained or tractable problem for registration. And I just want to sort of shout out to HipUnfold, which is fully available as a BIDS app. It can be used automatically for MRI data and can be customized to work with high-resolution data like this. And then finally, I also just want to show a quick representation of things we can do once we have combined multiple contrasts into an even more detailed brain model. So here I have unfolded maps of morphology averaged across our different samples, and then the big brain features. So basically the amount of staining that we see across big brain, which you can see, for instance, is very high in some subfields, very low in others. And then we can also combine that with the distribution of staining that we get, or, well, not staining, but intensity that we see in 3D PLI, which highlights different aspects of hippocampal architecture. And then we can take this multimodal information and do some dimensionality reduction. So in this case, I use a tool called BrainSpace, which generates these primary, you can think of them as components or gradients. The first one just naturally sort of very closely resembled our ground truth idea of where we thought CA2 should be. The second one very much for CA1, and then the third one here, sort of the very anterior and to some extent the posterior of the hippocampus, which are sort of homogeneous in structure in a way that they're very, very thin. So hopefully that sort of demonstrates the utility of building such a detailed model across different modalities. And yeah, so I hope that that illustration was interesting to you. I'd just like to acknowledge everybody else who contributed to this project. I also have a poster by the same name as this talk if you'd like to come by and chat. And I'd like to hear your questions. Thanks.
Okay, now we can take several questions. So is there any question from the audience? Yes, please.
Hi, Jordan. Really amazing work.
Thanks.
You showed there that the really high-res histological data on the unfolded space showed quite a lot of variability between the hippocampal subfield sizes and alignments. And I was wondering whether you think that's like a feature that is true of, to what extent do you think that might be affecting the alignment in the in vivo unfolding and alignment of the subfields? HipUnfold.
Just so I understand, is this the slide you're referring to?
No, the first one where you did the manual segmentations of the subfields and you showed that they don't look that similar until you do the multimodal alignment.
Oh, I see. So, okay, here, I'm just going to back up really quickly. So it's, I guess this is kind of what you're referring to, that the different subfields don't necessarily look identical in unfolded space. Mm-hmm. Yeah. We didn't necessarily expect them to be identical because we did this independently in native space for each of these two hippocampi, and they actually did end up looking fairly close to each other. So that kind of actually lends some support to the idea that a 2D topology is a nice way to represent the hippocampus to minimize intersubject variability. As for your question about in vivo work, currently the way that HipUnfold is set up, it doesn't do any registration in unfolded space. I'd like to develop that next. But yeah, hopefully that would improve the registration or the overlap we see between subjects even further.
Next question.
Did you assess neuropathology in your specimen?
No, as far as I know, these are two healthy, I think sort of middle to late age individuals.
Well, they are dead.
Yeah, of course,
so.
Yeah.
Well, in my experience, also these younger brains, they do show neuropathology, and I think it would be interesting to check whether that could explain part of the differences, large differences that you see in your shape.
Yep. That is kind of the direction we want to go. We want to, once we have these sensitive tools, we then would obviously like to look at different individuals. I should also say, though, that I'm not the first to observe this variability. There's actually a lot of people working on this question of folding within the hippocampus as well. And it does seem to be widely variable across different studies as well.
So just to add to that, I just recall a study about maybe 10 years ago by Carlos Cerati. He actually correlated, I'm not sure which way it went, in terms of the amount of folding was correlated with depression. It seems like depressed patients had either more or less folding. So that was, so it's interesting along these lines. It's like, you're trying to register the data and you're sort of normalizing for some information, but it's important to sort of be aware, right, of what information you're normalizing for, so that you don't throw anything out. It's interesting.
Yeah, exactly. Maybe it's also worth mentioning that this measure of gyrification is sort of proportional. It is a measure of gyrification. It's proportional also to surface area. So smaller surface area could mean a smoother hippocampus.
Yeah.
Other questions? Well, I have a question. Well, in PLI, yes, you also have this fiber orientation map in the native space, but is that possible in the future to also unfold and register fiber orientation information in the same space, or is that too complicated?
No, absolutely. We intend to do that. There is currently just an artifact with directional information in this data set, but we absolutely intend to do that. So I mentioned you can reversibly go between unfolded and folded space. You can do a transformation of that data to unfolded or even back to the original native space of 3D big brain.
Thank you. So anything else? Yes.
Should I get a mic? Thank you. I was wondering, what is the what are the implications of the chosen canonical form, which is a rectangle here? Or did you try ellipsoids and so forth to unfold?
Yeah, so you can actually create a shape that would minimize the amount of stretching that you would see between folded and unfolded space. That is possible to do. Aesthetically, I decided to go with a rectangle just because I find it's easy to orient. So up-down is anterior-posterior, and left-right is medial-lateral. I don't have a slide of it, unfortunately, but the minimal distortion shape actually looks a bit like a violin plot, where the anterior of the hippocampus is overall a little bit larger than the posterior.
Yeah, thank you.
Thanks.
So, okay. Yeah. I guess thank you very much. That was a great talk.
Thanks.
And that concludes our session for the day. Thank you.